Thank you. So I want to thank Shama and Bernardo not only for this invitation, but also for, during this really difficult time, dedicating a lot of work together with the companies to building and fusing communities. I think we really appreciate it. I hope we can give them a round of applause. So today, I want to ask the question, how do we learn from rare salient experiences? And so we know that in machine learning in the 90s, experience replay was one solution to rare samples where you could take all of the recent experiences, put them in a buffer, and then randomly sample from them over and over again to update the weights in your model. And this sampling improves efficiency. It breaks down the correlations between just sampling in adjacent samples, and it prevents catastrophic forgetting. But obviously, we don't care about all experiences equally. And so more recently, there was a set of updates where prioritized replay was a way of saying what are the important or unexpected or salient experiences, and can we essentially replay those preferentially. And in neuroscience, there's sort of equivalence to this, where neuromodulators that are sort of built to detect salient and unexpected things might tag the neurons that are active for reactivations that happen after the experience. Now, we know that there's analogs to some of what I just described happening in the hippocampus through decades of work. But my lab is interested in sensory learning. How do we learn about the visual world? And so we wondered whether or not similar features were happening in visual cortex. And so the first part of this talk, I'll describe this dialogue between the visual cortex and the hippocampus in mice. And then this dialogue, you can think of it very simply. So you have the cortex and the hippocampus, and then you have the thalamus, which is bringing in some of the information from the visual world. During active presentation of stimuli, the thalamus is really dominating the dialogue with cortex. But in the moments of quiet waking, the thalamus seems to be suppressed or silent, and this facilitates a sort of a predominant dialogue with the hippocampus in cortex and a dominance of sort of recurrent activity in this network. And we think that this is important for learning in ways that I'll expand on. Now, in the second part of the talk, I'll then talk about the structure of individual moments of offline replay in the cortex. And these replays of recent experiences we think are important for actually shaping neural activity, driving plasticity in the network. So to begin with, I want to highlight, especially for the trainees here, like an amazing experience I had in a study that started at the beginning of the pandemic and that tried to give us intuition on how the dialogue between cortex and hippocampus was happening. Really, people didn't know very much about this, but there was this amazing open source data set from the Allen Institute where people recorded with very dense neuropixel electrodes from really hundreds of neurons at the same time, and they recorded throughout 6 different visual cortical areas, as well as the hippocampus and the various parts of thalamus. And this was a great opportunity for sort of thinking about different parts of the brain and what they're saying to each other in different states, aroused, unaroused, and so on. And so these data sets had many different visual presentations to the mouse and lots of recordings, but they also had a 30-minute period where the mouse was just awake and not doing very much. And so what Huijang here noticed during this time, this was a collaboration of Huijang Zheng with Minwan Jung and Vijay Nambaviri, is that there are different parts of the hippocampus, and this is sort of this C-shaped loop here, that show these 100 hertz wiggles called ripples, and these ripples reflect the concerted activation of thousands of neurons. And each of these ripples, as Jan Drugovic and others have shown, really are moments where there seems to be an episodic kind of memory type replay of a previous experience. But you'll notice that different parts of the hippocampus, which have been associated with different cognitive maps of the world, don't show these wiggles at the same time. In fact, sometimes you have a dorsal hippocampus wiggle, and sometimes you have an intermediate hippocampal wiggle. And so we said, what is going on in the cortex while the hippocampus is in this sort of memory-related state. And what we found was that, for example, in this neuron shown over here, each of these rows is a set of spikes with each of these dots, and they're aligned on the moment of a hippocampal ripple. And you can see that this cortical neuron is coactive when there's an intermediate hippocampal ripple, but not when there's a dorsal hippocampal ripple. So it seems to be talking in a privileged way to one region of the hippocampus. And if you look at this now, where each row here is basically a heat map of the same data, but for a different neuron, I mean, we would never be able to collect these many neurons ourselves, but using this amazing data set, you can see that there's some neurons that are preferentially talking to one part of the hippocampus, the intermediate region here, and then there are other neurons, sometimes neighboring neurons, that are preferentially talking to another part of the hippocampus. And it's mostly happening during quiet waking. So that doesn't really happen anymore in modern society because of iPhones, but you can imagine that mice still have this privilege. What we were surprised to see is that actually a full quarter of all the neurons in the cortex seem to have this dialogue with the hippocampus during these ripples. And it didn't matter if you're in an early sensory area or in later higher order areas, they were all talking. And this was true across layers of the cortex as well. And so just to summarize, we found that actually we found different ensembles of neurons that are defined by whether they're talking to one part of the hippocampus or another. And these different ensembles of neurons in cortex seem to carry almost identical carbon copies of the visual world. They basically encode the same distribution of visual stimuli, but they're relaying this information, we think, to different parts of the hippocampus. And we speculate that these different ensembles may then bring new visual information to be incorporated into different cognitive maps. The most important takeaway from this study is that during a moment of hippocampal ripple, shown here at time 0, some of these cortical neuron ensembles are increasing their activity, but importantly, the thalamus, which we could record at the same time, is decreasing its activity. And so this really highlights this moment of high recurrence where the cortex is actively sending and getting back information from the hippocampus. And so you can think of active waking and then quiet waking and then these moments of hippocampal ripples as particularly sort of offline states where there's really a predominance of strong recurrent inputs as the dominant drive to a given neuron. This will be important later. And we hypothesized that during ripples and associated offline reactivations, so reactivations here I just mean a bunch of neurons get activated by stimulus and then offline that same pattern of neurons gets reactivated. We think that these reactivations are somehow helping to incorporate a new stimulus representation into the model of the visual world. We wanted to now get at the actual structure of individual reactivations, the pattern of neurons that are activated by a stimulus and then reactivated offline. During these reactivations, we know that these neurons are not really very loud. They will fire 1 spike or 0 spikes. And so you really need a lot of neurons in order to be able to identify and understand the structure of these events. And so we had done previous studies where we had complicated tasks with cues and rewards. But Nia Nguyen, when he came to the lab, took a simpler approach. And he said, why don't we just study the evolution of responses to the same simple visual stimuli in the absence of any rewards or punishments? And let's just watch what happens when we present the same stimulus over and over. What Nia did, and these are what the stimuli look like, but the important thing that Nia did that was different than all previous studies of just passive presentation of a visual stimulus was that he gave about a minute between each stimulus. Lots of time for the animal to just tune out, and that's when we could study these reactivations. And we presented these 2 stimuli interleaved for several hours. So very simple, but importantly recorded from many different brain areas. This is a top-down view of the mouse brain. These are all visual areas that respond to various visual stimuli. We're using 2-photon calcium imaging to record up to 7,000 neurons, and we do that also by recording across different planes. The only important thing from this slide is that if we got rid of 9 out of every 10 neurons, most of the results I'll show you would go away. So this was a rare moment where actually a rare situation where we really needed a lot of neurons in order to actually learn what we needed to learn from the data. So what I'm showing you here are a subset of those neurons, the ones that are preferentially responding to one of the stimuli, S1, or to the other stimulus, S2. And you can see one thing already is that in the first trial of the day, both sets of neurons are responsive to a visual stimulus, S1. But by the end of the day, there's a sharpening. Only some of the neurons are still responsive and the other neurons are less responsive. And so you can think of these population responses as sort of moving away from each other over time for these two stimuli. And this moving away from each other, we can capture that by just looking at a running correlation of the patterns of responses to the first stimulus and to the second stimulus. And so this correlation is going to go down and down and down as these stimuli and their representations become further apart. Okay, so where do reactivations come in? So what I'm showing you now is a presentation of a stimulus for 2 seconds, and then just looking at the data for the next minute, you can see that there are these moments of sharp activity that's synchronous across many neurons. And after the presentation of this S1 stimulus, we see S1 neurons reactivating. But after the presentation of the S2 stimulus, the other stimulus, we see another pattern of neurons that are reactivating. So it seems like there's more reactivations that are matching the most recent stimulus. And in the beginning of the day, there are many more of these, and I won't go into that. But I can use a simple logistic regression classifier to identify the moments when these reactivations are happening. And what I want you to notice is that when we present one stimulus, each of these rows is a trial, we see lots of these red dots, so lots of reactivations of that stimulus, but very few reactivations of the other stimulus. So it seems that even a minute after the mouse has seen something, it's still thinking about that stimulus and not the other stimulus. And likewise, for stimulus 1, you see more reactivations of stimulus 1 and not stimulus 2. So this is exciting because it really suggests that what we're picking up isn't just random noise, it's actually content that's related to something that happened a minute ago. And these reactivations are very common at the beginning of a session when the mouse hasn't really seen the stimulus before or hasn't seen it in many days, and then the reactivation rates decline over time. That'll be important because we can think of the reactivation rate as kind of a learning rate. And also the probability that you see reactivations of a stimulus is about 3 times higher for the most recent stimulus than for the other one. So this really is a very strong bias to recent salient events. When we silence the cortex, so the mouse is still seeing the stimulus, is coming in through the eyes, it's passing through lots of other brain areas. The local regions we're imaging have been silenced, so they can't get activated by that stimulus. We silence for 2 seconds, we stop the silencing, and now for the next minute, we don't see any more these reactivations. So we think, similar to in Carol's talk yesterday, the activation of a neuron is actually important for whether it's going to participate in these offline reactivation events seconds or minutes later. Now, to bring this back to the first part of the talk, we, in some heroic experiments from Andy, Kelly, Arthur, and Oren, were able to combine imaging of all of these neurons with simultaneous recordings of hippocampal ripple activity. And what we could see is that whenever we identified these events in cortex, they were coupled to ripples in the hippocampus. And we think that essentially the entire cortex, probably different parts of it, have been attending to different important things. And all of the neurons associated are now reactivating together in the same 200 milliseconds with the hippocampus as a conductor. So now I'm going to get back to this idea that the stimulus responses are separating over time, and this might be useful based on previous work for the ability to then discriminate different objects as the representations become more orderly and separable for these two repeated stimulus presentations. And what we can see at the level of different neurons is that the cortex does a really good job of maintaining the same average response to a stimulus. We think this is important as a kind of circuit homeostasis to keep the circuit generally responding in a similar range. But it does this by having some neurons decrease their responses to the same stimulus, but having an equal number of neurons increase their response, these maroon neurons, to the same stimulus, so that the average population activity is the same. And so this is the main finding of this work that Nia observed. So what I'm showing you are 6 dots, and the size of the circle is the magnitude of the response to this first stimulus of the day. So this is a specific set of neurons, and they're responding to this one kind of stimulus, S1. And now we can find a pattern that happens during a reactivation. And you can see that these reactivations are mostly the same pattern as the stimulus itself, stimulus response itself, but slightly different. Some neurons are activated a bit more during the reactivation than during the stimulus experience itself, and others less than expected. But this isn't immediately driving plasticity in the sense that half a minute later, the next presentation of that stimulus, you basically have the same pattern. However, an hour later, actually, the pattern gradually shifts so that some of these neurons are now being driven by a stimulus more, and others are being driven less. And so this plasticity in the network, an hour later, is predicted by these offline recurrent, high recurrence moments in the first 5 minutes of the session, which was really surprising to us. We could predict future changes in neural activity from just concentrating on these offline states. And so to quantify this, if you think about the neurons that are increased neurons that I defined here, those increased neurons we predict have greater participation in these reactivations than in the stimulus response itself. And so we can take the difference between the reactivation activity and the stimulus activity. And basically, just in a nutshell, that's what I'm showing here. Increased neurons tended to over-participate in early reactivations, and decreased neurons, the ones that subsequently decreased their responses, tended to under-participate in these early reactivations. I know that's a mouthful, and I'm going to plot this for you in a different way. So, as I told you, there's an evolution of the response to the same stimulus over the course of the day in these 7,000 neurons. And we can reduce that to a simple low-dimensional space, and each dot is the response pattern on one trial. And so these dots are going to move in this space because the pattern is changing over time. And so we can project the activity across this dimension of movement, and we can derive a scalar for each trial, for each pattern we see on each trial. And as expected, this pattern is changing. It changes fast at the beginning of the day and slower at the end of the day. So if the reactivations were carbon copies, then you'd imagine something like this, which is the reactivations, when projected onto the same axis, should also slowly change over time, but that's not what we saw. Instead, what we saw was that the reactivations were already different than the stimulus responses, and they were predicting where the drift was going to happen, where the changes in this population was going to happen over time. We saw this for both stimuli, and we saw it across many individual trials in mice. Now, one cool thing about being able to track the same neurons across days, which we did with the help of Bernardo Sabatini's lab, is we could look at this drift that we see, and then we could also study it across many days in the same neurons. And so if you think about the movement projected onto this change in activity in the first day, so that's basically what I'm showing you here. It's as we expected. You have movement of the stimulus response, but no change in the reactivation pattern. And then on the second day, there's a bit of a kind of a backslide. But eventually, this movement along that particular axis in neural activity space is flat. But we can also look at the other, there might be movements across other directions in the space on other days. And so if we look at the difference between day 1 and day 6, actually you see that the stimulus response is continuing to change over time. And even across days, this can be predicted by these offline reactivation events. So just to kind of summarize this graphically, we think that we present two kind of unnatural stimuli, and the response patterns are different, but they're partially overlapping. And then over different trials, the response patterns get further and further apart, which might be useful for discrimination. But we can predict where they're going to go, because the reactivation patterns early in the session are already far apart, and it's as if they're dragging the stimulus responses towards them. Now, this idea of actually having almost a knowledge of the activity patterns that you want the network to move towards and then apply plasticity rules afterwards is something that's also come up a lot in deep learning recently. Some people call it target learning, other people call it prospective configuration. But the basic idea is that it's an interesting, effective, and biologically plausible alternative to backpropagation that uses energy models to essentially say, if you can predict where you want to be early in learning, then you can gradually adjust ways to get there in an efficient way. And one of these papers by Benjamin Grev's lab shows that indeed, if you in slices record from a single neuron, and you can stimulate its basal dendrites, which are where the feed-forward inputs are coming in, or the apical dendrites, where the feedback recurrent inputs coming in. When you stimulate, and just look at my cursor here, very strongly the recurrent inputs together with the basal inputs from feed-forward, when you post-stimulate these, you actually can potentiate the feed-forward inputs. So this you can think of as you've essentially got these reactivations, and the neurons that are active during those reactivations are training their feed-forward inputs so that the next time a stimulus comes, it's going to drive those neurons more. So if this is true, then in our model, the reactivations alone should be enough to predict this change in drift quantitatively. And so we used a very simple toy model where we just took the first stimulus pattern of the day, a single trial. And then after that, all we did is we looked at the reactivations that we observed in our data. We said, are those reactivations enough if we just propagate a simple rule to predict all the subsequent stimulus patterns and how they change over the rest of the day? And so this is just a very simple rule with a single parameter, plasticity term that we fixed across animals. And what you can see is that it captures this really nicely, this change in the stimulus. It actually captures the change in responses in these neurons, not just over an hour, but can even capture like every 5 minutes, fast changes in response patterns as well. So these instantaneous offline moments might have a lot of plasticity potential, and we're interested in trying to perturb them selectively. But they did more than that. They actually predicted things that we didn't even train at all in this model to do. So I told you before that there's a decorrelation of these two response patterns. If you look at the data just for these neurons that are decreasing their response over time, you see that those neurons that are decreasing their response actually are the ones that are driving this orthogonalization or separation of the two response patterns. And in the model data that's just using the reactivations to update the stimulus, predict the stimulus response, we see all of the same effects, even though we didn't actually train the model to create any of this. So that was really surprising to us. And overall, I think that suggests to us that there may be a lot of value, especially when you're in regimes where you have lots of sort of offline time, but not a lot of sampling of the stimulus, to use these reactivations to gradually update. And I think one thing I want to highlight is the more reactivations you have, the faster the learning rate. And so salient experiences, as I'll show you in the next slide, actually drive more reactivations, and therefore there's more updates perhaps and more rapid learning. What I just said was that when you have a stimulus that's preceded by a different stimulus, you have a slight increase in reactivation rate. And the stimuli at the beginning of the day drive much higher reactivation rates than at the end of the day. And so there's a model that is similar to what Karel Svoboda introduced yesterday, a three-factor model where you might have Hebbian plasticity, but facilitation in a way through a neuromodulator that is carrying information about what is novel and what was surprising to the animal to enhance perhaps the plasticity. And one way this might go is you have a stimulus, it drives calcium release from a bunch of neurons, and it also drives norepinephrine release, or it might be other neuromodulators. Importantly, we know that this in cortex is driving second messenger signaling. And I think just getting at the question of mechanism that Conrad brought up, one of the exciting things for studying second messenger signaling downstream of neuromodulators is that when we think about spiking activity of neurons, we have no idea if the plasticity is in those neurons or if it's being inherited from other neurons. But when we think about local changes in intracellular signaling, this really suggests that there might be a plasticity update happening in the neuron we're studying, which is, I think, an exciting way to get closer to mechanism. And so we predict that these elevations in a second messenger, like cyclic AMP, might be elevating excitability of the neurons or some of their dendrites, and this would allow those neurons to then be more recruited during reactivations offline, which might explain our decline in reactivation rates over time. And so just to kind of sort of describe our efforts in this direction, we have done in the lab, with the help of Bernardo's lab, several studies in different parts of the brain where we have worked with the Naga lab to develop methods both for tracking this second messenger, in this case cyclic AMP, and also for manipulating it and for optogenetically perturbing it upwards or for blunting it. And we can take this set of works and bring it to the cortex and think about what happens when this third factor, in this case, norepinephrine axons that are coming to the cortex and that are bringing a surprise signal into the cortex. So what happens if we activate those axons and release the norepinephrine? What do we see downstream? So even if the surprise that happens for 1 second and those axons get activated for 1 second, when you record the cyclic AMP, the second messenger levels within these neurons, you see that that lasts for actually 30 to 60 seconds. And this is important because it kind of also gives us a sense of like things like eligibility traces perhaps, or ways in which something that happened 30 seconds ago might actually be influencing the network 30 seconds later in a way that's kind of invisible if you're just looking at spiking activity. And you can see that this is true even at the level of individual spines. Some spines, if you stimulate the release of norepinephrine, you get an increase in cyclic AMP, and in neighboring spines, you don't. So if anybody is interested in this general direction of reactivations, there's a very talented, theoretically minded experimentalist in my lab. He's a graduate student. His name is Josh Stern. He's a new member of the Kemper Institute, and I'd really encourage you to look him up and to talk more about possible collaborations. And I just want to thank everybody who helped with all of this work, the current members of the lab, my family, my collaborators, and my sources of funding. And I left a bunch of extra time for questions, so thanks for your time.
Very nice talk. But novelty can be represented even in V1. So how did you distinguish this with novelty representation? So basically the novelty should be probably represented only when you perceive, but not really the reactivation. So how did you distinguish the novelty representation with this your model?
Yeah, so in our data we see that there is a slight increase in neural responses to novelty, and then we also see an increase in reactivations after a trial where either the mouse hadn't seen the stimulus that day or hadn't seen the stimulus in many minutes. So I think both of them can be true at the same time.
The representation itself can be different, so that the novelty can be also representation, not only the amplitude or the frequency.
Yeah, so what was really sort of interesting about having our stimuli 1 minute apart, so the question is, well, there's complexities where if you have stimuli that are happening in rapid succession, actually you'll have adaptation and you'll have a rapid decrease that stays over many trials in the magnitude of the response. But when we actually have the stimuli a minute apart, what we were surprised to see is that we don't actually see that at a population level. There's just as many neurons decreasing their activity as increasing. But the reactivations are happening much more frequently after the novel stimuli. So we're still trying to figure out exactly how that works. Thank you.
Question with the reactivation thing. It's kind of mysterious why it happens. I think the target learning explanation is a little unsatisfying because it's not clear to me why you'd want this arbitrary neural pattern to be target. Do you think it could have something to do with trading off perception and memory? And in particular, the more you see and experience something, the less work you have to put in to perceive it, and you can just kind of remember how it looks like before.
Yeah, so I mean, thank you for allowing me to speculate. I think And the stimuli that we're presenting are very unnatural. And one way that you can think about how the visual cortex wants to represent stimuli is that it's been trained with a huge battery of natural statistics over the lifetime of the animal, and that there may be some low-dimensional manifold that essentially contains all of the responses. And this manifold might actually have good properties, like it's naturally orthogonalizing responses to different objects. And now you come in with an off-manifold kind of artificial stimulus, and the cortex wants to bring that back onto a natural manifold, maybe because it also has good readout properties downstream. And so the question of why you might want to actually have this drift towards that manifold, that's one possible solution. And one way that we could test this in future is to come up with artificial stimuli that would already sort of push the responses onto the manifold and then see if there isn't any further drift. So I think that answers your question, but if it doesn't, let me know. So the question of why do we, or how do they happen, or maybe you can ask again.
I mean, could you comment on the perception memory trade-off thing? I don't know if it's true, it's just a thought.
Yeah, so I mean, essentially, if you believe the sort of the predictive coding view of what the cortex is really doing, the things that we perceive most saliently are the things that we really didn't expect, either because we didn't expect them in time or because we didn't sort of have a model that they're going to come. And so I think in that case, those are the stimuli that we perceive. Those are also the stimuli that then we want to then keep mulling over in an offline moments so that we can improve our model and improve our prediction of those stimuli in the future. That's kind of how we think about it. Whether or not those offline reactivations are themselves instances of conscious imagery, there's indications from recordings of these ripples in cortex of humans and in hippocampus at the same time that suggest that the same phenomenology we're seeing is happening in humans that are asked to do a voluntary recall. So it could be that the mice are actually doing this over willfully.
Dominance columns and orientation columns, and the eventual learned patterns that are evoked slide into the things that you already see spontaneously early on.
Yeah.
Do you think that these things are the same? And one of the possible reasons for having those structures initially is just pre-built wiring, that you have shift, twist, symmetries, and things like this. Is this going to happen more generally?
Completely. So right now what Josh that I introduced is doing is he's looking at activity before we've ever presented any of these stimuli, and he sees patterns of neural activity, and you see this all the time when you look at spontaneous activity, these flashes and other kinds of patterns. And these patterns are, we think of them potentially as core elements that then can be combined in various ways to be assembled during these reactivations in the service of associational learning. So if you have these two different patches that never really happen together, and now you start seeing them happening together on the screen all the time, are there sort of primitives in the assemblies that you already have inherent, built into the network, sort of neighboring cells that neighboring parts of space, they tend to be connected to each other. Now you can essentially have that flash of cells and another flash of cells encoding a different part of space. Now they're happening together in the same 200 millisecond window, and it's easier to then have plasticity rules to then bind those two assemblies together. That's what we think is happening, and we're testing.
Thank you so much for the talk. I was really curious about manipulating the time decay in the reactivations, or any way in which you can mess with this. And one way in which I was thinking potentially is adding another type of neuromodulation, maybe instilling fear. I'm wondering if the decay is actually increases, right? Even if the reactivation goes away much faster or much longer. Can you mess with that? What happens with people with PTSD, for instance? That reactivation, is it more clean?
Yeah,
So
such a great question. I mean, we've been trying to manipulate these reactivations online, and it's been really challenging for reasons that I won't bore you with. But there are new tools now that are optogenetic means of rapidly and transiently blunting cyclic AMP or PK activity. And we think those will be really effective in terms of being able to sort of prevent plasticity in an intra-trial interval without messing with the next stimulus presentation. But doing this also by presenting kind of competing streams of appetitive and aversive stimuli is a good idea. Thank you.
You started to touch on this a little, but does this process of recall, does that correspond to like the human default mode network kind of actively recalling these things, or is it happening more unconsciously just on a neuronal level? And is the reactivation, that increased sensitivity, is that kind of like the learning of priors?
So, yes, so I mean, we think that first of all, there is a lot of reports of sort of imagination and imagery during the default mode activity that is observed in humans. And we do think that these ripples can happen both during quiet waking when there's mind wandering and during sort of deliberate willful recall of stimuli. It has to be dissociated from the cue, so it has to be sort of self-generated. And in both those cases, I think these ripples are really just indicators of a kind of communication that might be important for bringing back into the buffer a recent experience. And the second part?
Oh, if the increased sensitivity was a learning of priors.
Yeah, so that's definitely our hypothesis right now, but I think it's really just that at the moment. Thanks.
Thank you.
Thank you. Follow-up question on the dynamics of the decay reactivation. I'm presuming that on the following days that it's exactly the same stimulus being shown. So in that case, when in the decay, after there's no more activity, how fast does it come back up? And does it ever come back to the original level or not?
Yeah, so on subsequent days, I think there's sort of a temporary forgetting, and so you do see reactivation rates go up again, but by the 5th or 6th day, there's definitely less of that. So presumably there's sort of both fast learning within a session, there's sort of a partial unlearning in between sessions, and then over several days, this sort of becomes locked in, and there's less of that. Now, importantly, the sort of the ripple rates that we and others have measured during learning stay constant. It's just that the choice of what is reactivated and whether it's related to this task or probably like the terrible smell of a human holding the mouse for the first time, at the beginning of the next session, those might be reactivated instead.
Thank you.
It was fairly striking the difference in time scale of the ripple itself and the reactivation you see in Cortex, which seemed to precede it by like a second or something.
Yeah.
Can you speculate on why that might be?
Yes. So, I mean, when you do the same kind of thing in low Frank's data where they sort of do an analytical task, you see very similar things. So one speculation is you have an experience, the cortex is now biased to kind of processing that experience. And so how do you actually then bring in sort of your prior model or linking? One way is that the cortex is now biasing the content of what's happening in the hippocampus. Hippocampus is naturally orthogonalized. And so the activity goes there and then triggers an indexing of additional neurons in the cortex that are now, part of its natural manifold. So there's a pinging from cortex to hippocampus that biases the content. The hippocampus then in return activates the cortex, but not the exact same neurons. Now it's the neurons that are already part of the internal model.